Good afternoon, ladies and gentlemen. My name is Dimakatso Gumede. I will be giving a presentation entitled Making Macrophages in a Dish, Modeling the African Response to Immunity. The inflammatory response is a critical process for clearance of microbial infections and wound healing following tissue damage. It is a process that requires strict regulation to prevent systemic inflammation and organ failure. Now, this process of systemic inflammation is also observed in cases such as COVID-19, where, for example, the virus enters the epithelial cells of the lungs, inducing an immune response and activation of immune cells such as macrophages that start increasing the expression and release of inflammatory cytokines to fight off the infection. But that could also lead to the tissue damage and activation of the T-cell adaptive immune cells that also amplify the release of the cytokines, causing what is also known as a cytokine storm. Now, there are different models that have been established for modeling different diseases, and the first one is the simplistic one where we call the two-dimensional cell culture modeling. And there's also the use of animal models such as mouse models, but those do not faithfully recapitulate what we call a phenotype that is observed in humans. Therefore, the use of what we call the mini organs or the organoids is what is used now to model those diseases and to actually see the phenotype that is observed in humans. One of those will be to either take stem cells from different tissues or the stem cells from a blastocystis. And what is now used is the use of skin fibroblasts and reprogramming them to pluripotent stem cells and then creating those mini organs or organoids to mimic a specific tissue or a specific organ. And that is what we are using in our lab. And with that, showing just an example of what an organoid looks like. It's an intestinal organoid that has been infected with the SARS-CoV-2, highlighted in white or displayed in white, just showing how the virus enters the organoid. And these models help us to have a clear understanding of the processes that occur in response to the virus in different tissues. Now, our aim is to mimic or to engineer such models of immunity in our lab in order to identify therapeutic targets for systemic inflammation. In our lab, we use the induced pluripotent stem cell platform, where we use human fibroblasts, skin cells, and reprogram them with a virus, a non-integrating Sendai virus, to make pluripotent stem cells that can become any cell type in the body, including the haematopoietic cells. And those cells can then be edited or engineered to either correct or induce a disease model using the CRISPR-Cas9 system, and then further on differentiating them to those specific cells in order to understand the mechanisms that are involved in disease occurrence and how to induce the therapeutic. For our research, we'll be using the human iPS cells, those are the induced pluripotent stem cells, and differentiating them into the macrophages, which are part of the haematopoietic progenitor cells. Our research approach for that is to create what we call the gene-edited stem cells using the CRISPR-Cas9 system, and then differentiate them into the iPSC-derived macrophages, which will then induce an immune response in order to determine the mechanisms that mediate the reduced immune responses that can be used to mitigate the systemic inflammation that is observed in different diseases, including COVID-19. For our research platform, this is just an image just showing you what we look for in the reprogramming and the differentiation of our stem cells, where they become what we call the monocytes from the pluripotent state, and what they become macrophages once we take them through that differentiation process. Furthermore, we show that they can stain and show the feature and the phenotype of the normal monocyte or human monocyte cells. Our model can also be used for HIV models, and which is what we have also done in our lab, where we've shown that the normal cells that still contain this CSIR5 receptor that the HIV uses to enter the cells, once it enters the cells, it causes those macrophages to start clumping up and dying in what we call syncytia. And knockout of the CSIR5 receptor actually prevents the viral entry into the macrophages, thereby showing healthy phenotype in the image below. So what we're working on is creating those disease-in-a-dish macrophage models, addressing the systemic inflammation that is caused by microbial infections, including TB and SARS-CoV-2. And this will also help us to observe the response and how to target those mechanisms to prevent the immune activation that's also observed in HIV-positive individuals, and also help us to identify those therapeutic targets that will modulate the inflammatory response in the African context. That brings me to the end of my presentation. I will now take questions.
That's it.